14,310 years. That is the radiocarbon date returned by accelerator mass spectrometry on charcoal recovered from a cave in southern Yunnan Province, China. The same layer that yielded a human skullcap with thick brow ridges, a low forehead, and a partial thigh bone so small and so primitive in proportion that it more closely matched the femora of Homo habilis, a species thought to have vanished more than 1.5 million years earlier than that of any modern human alive today. The bones were excavated in 1989, then sat untouched in a Chinese provincial museum for more than 2 decades. When an international team finally examined them, the discovery split the scientific community in two. One side argued the remains belonged to a previously unknown archaic species that had survived in the mountains of southwest China until the dawn of agriculture. The other side argued they were modern humans whose unusual features had been overinterpreted. In 2022, ancient DNA appeared to settle the question. Then, in 2025, a team at Harvard reopened it. This is the story of the Red Deer cave people, and of a mystery that ancient DNA has not, in fact, finished solving. The first clue came in 1979 when a Chinese geologist named Bao Zhende, working in a cave near the village of Longlin in southern China, removed a consolidated block of sediment containing a partial human skeleton. The block was so densely cemented that the skeleton remained encased in rock for the next 30 years. In 1989, the same researcher led a separate excavation about 300 kilometers to the southwest, near the city of Mengzi in southeast Yunnan Province. The site was called Maludong, Red Deer Cave. The team uncovered fragmentary remains of at least 3 individuals, along with stone artifacts, deer antler awls, and lenses of charcoal from ancient hearths. The Maludong material was published briefly in Chinese journals and then set aside. For nearly 2 decades, almost no one outside China examined either collection. In 2008, Ji Shuiping of the Yunnan Institute of Cultural Relics and Archaeology partnered with Darren Curnoe of the University of New South Wales to reinvestigate the Maludong material. Accelerator mass spectrometry. Radiocarbon dating placed the human remains between 14,300 and 13,500 years before present, the very end of the late Pleistocene, tens of thousands of years after every other archaic human population on Earth was believed to have gone extinct. In March of 2012, Curnoe, Jai, and 16 co-authors published a long technical paper in the open access journal PLOS ONE. The Maludong skullcap displayed a mosaic of features. The vault was relatively high and rounded, consistent with anatomically modern humans, but the frontal bone was unusually thick, and the brow ridges were robust. The forehead sloped backward more steeply than in modern populations. The mandibles showed strong alveolar prognathism, with the lower face projecting forward in a way more typical of earlier hominins. Several molars displayed toroodontism, a feature associated with archaic populations including Neanderthals. The long-lean one skull, reconstructed from the 1979 block, was even more unusual, combining a relatively modern brain case with a pronounced brow torus, a flat mid-face, and an extremely robust mandible. The authors offered 2 interpretations. The first was that the remains represented the late survival of an unknown archaic species. The second was that they were hybrids, the products of interbreeding between anatomically modern humans and an unidentified archaic group whose own fossils had not yet been found in mainland East Asia. Either interpretation carried a radical implication. For the period from 15,000 to 11,000 years ago, the textbook view held that Homo sapiens alone walked the earth. The Red Deer cave material, if accepted at face value, meant that humanity had shared the late Pleistocene landscape of East Asia with at least one other archaic population. The most provocative finding appeared 3 years later. In December of 2015, Curnoe and his colleagues published a follow-up paper in PLOS ONE describing a partial femur from Maludong. The shaft was unusually narrow. The walls were buttressed in a pattern more typical of Homo habilis and early Homo erectus. The neck was elongated. The reconstructed body mass of the individual was approximately 50 kilograms, extremely small for any Pleistocene human, modern or archaic. The Maludong femur, the authors wrote, was the youngest fossil with archaic morphology, yet found anywhere in the world. The research community responded with a mixture of intense interest and serious skepticism. Without ancient DNA, the question could not be resolved. By the mid-2010s, the question had taken on much greater significance. In 2010, the genome of a previously unknown human population, the Denisovans, had been recovered from a single finger bone in Denisova Cave in southern Siberia. The Denisovans had been present in Asia for hundreds of thousands of years. had repeatedly interbred with the ancestors of modern Asians, and had left almost no fossil trace. If the Denisovans could remain so nearly invisible despite their genetic legacy, the possibility that another archaic population had persisted in the mountains of southwest China until 14,000 years ago could no longer be ruled out on prior probability alone. Some researchers began to wonder whether the red deer cave people might be late surviving Denisovans, or hybrids carrying a substantial Denisovan inheritance. Resolving the question required ancient DNA, and ancient DNA from southern China, with its hot, humid climate and acidic cave soils, was almost impossible to obtain. Then, in July of 2022, a team led by Xiaoming Zhang at the Kunming Institute of Zoology of the Chinese Academy of Sciences published a paper in the journal Current Biology announcing that they had succeeded. The paper reported for the first time ancient DNA recovered from one of the Maludong individuals, the skullcap given the informal designation Mengzi Ren, abbreviated MZR. The headline finding was that MZR was an anatomically modern human. The 14,000-year-old woman whose skull had been pulled from the Red Deer Cave belonged to maternal mitochondrial haplogroup M9, a lineage estimated to have arisen between 47,000 and 50,000 years ago. Her nuclear DNA showed deep affinities to ancient and modern populations of southern East Asia, with no detectable contribution from Neanderthals or Denisovans beyond what is found in living East Asians today. There was no archaic ghost lineage in her genome. The Zhang team also reported that the MZR genome shared a significant portion of its ancestry with the deeply diverged East Asian population that contributed to the founding ancestors of the first Americans, suggesting that her population was descended from, or closely related to, the source group from which Native American ancestors had separated before crossing into Beringia approximately 20,000 years ago. For paleoanthropologists, who had spent a decade arguing about whether the Red Deer Cave people were a new species, the answer, at least according to the MZR genome, was no. They were modern humans whose morphology had been misread. The recovery itself was a substantial technical achievement, drawing on approximately 30 to 50 milligrams of bone powder under strict anti-contamination protocols. But the yield was modest. Total endogenous human DNA represented only 11.3% of the recovered fragments. The remaining 88.7% was bacterial, fungal, and environmental contamination. The paper was widely covered in international media, with headlines declaring that the Red Deer Cave mystery had been solved. It had not been solved. Two earlier findings had already complicated the picture. In June 2021, a team led by Tianyi Wang and colleagues published a major paper in the journal Cell that included the genome of the Longlin 1 individual. The Longlin genome, like MZR, was anatomically modern but genetically distinct. Despite living in caves only 300 kilometers apart and roughly contemporaneously, the two individuals did not appear to belong to the same population. In February 2021, a separate group led by Sun and colleagues published a paper in the Proceedings of the National Academy of Sciences applying uranium-thorium dating to deer bones and cave flowstones at Maludong. Their analyses indicated that some of the deer bones from the human-bearing strata were in fact Middle Pleistocene in age, far older than the radiocarbon dates on charcoal had suggested. The implication was that the Maludong cave deposit had been disturbed at some point in its history, mixing fossils of very different ages within the same apparent stratigraphic layer. The authors recommended that the geological age of the human remains be regarded as uncertain until they were directly dated. In October of 2024, with a final published version appearing in Current Biology in March of 2025, two researchers at Harvard Medical School pushed the question further. The lead author was Daniel Tabin. The senior author was David Reich, head of the largest ancient DNA laboratory in the world. The Tabin and Reich critique was technical and devastating. When the Harvard team examined the publicly released MZR sequencing data, they found error rates many times higher than those typical of well-preserved ancient genomes. The pattern of errors was abnormal. They did not follow the characteristic damage signature of authentic ancient DNA. When the Harvard team ran standard contamination estimation tools, they estimated that only 55 to 66% of mitochondrial reads matched the consensus sequence, far below the figures reported by the Zhang team. The Harvard team concluded that the published MZR sequences appeared to contain substantial contamination by modern human DNA. Even setting that aside, they could not reproduce the population genetics finding that had generated the most attention, that MZR was equally related to ancient Northeast Asians and to the source population of the first Americans. When the Harvard team analyzed the same data using independent methods, the special connection to Native American ancestry largely disappeared. If Tabin and Reich were correct, the headline conclusion of the Zhang 2022 paper could not be reliably drawn from the published data. The genetic identity of the Red Deer Cave people remained, in their assessment, an open question. That question is still open today. If MZR and Longlin 1 were modern humans, as both genome papers initially suggested, then the unusual morphology must reflect either extreme variation within late Pleistocene Homo sapiens of southern China, or population-level retention of robust features in a small, isolated group. Neither possibility is well documented elsewhere in the East Asian fossil record. If the genome data are contaminated, as Tabin and Reich argue, then the original archaic survival hypothesis returns to the table, and the hybridization hypothesis returns as well. So does the possibility that some of the human remains are substantially older than 14,000 years and represent a much earlier population. What remains certain is the physical evidence. There are bones in Yunnan and Guangxi that look unlike the bones of other late Pleistocene East Asians. There is a femur whose proportions match no known modern human. There are skulls whose mosaic of archaic and derived features has been described and redescribed by careful researchers for over a decade. They are objects with measurable dimensions, and they require an explanation. The next step is direct accelerator mass spectrometry radiocarbon dating of the Maludong and Longlin human bones themselves, paired with new ancient DNA extraction attempts using improved tropical climate protocols. Until those are completed and replicated by independent laboratories, the question of who exactly lived in Red Deer Cave at the end of the last ice age cannot be answered. Our genetic past is not settled science. It is an active, unfolding investigation, and at Maludong, 14,000 years after the last fires were lit and the last red deer were butchered against its walls, the investigation is still in its early stages.